here. So, so Professor Miguel Weil from Life Science Faculty, discovery of cell-based phenotypes for personalizing ALS, drug screening and therapy. Please.
Okay, thank you very much for having me here. And I want to thank Vivian Rory for the excellent talk and for the great introduction about the disease. So it saved me a lot of time introducing the disease for you. And I will talk on how we found probably a way for personalizing ALS drug therapy. And this is based on cells coming from patients. We isolate phenotypes, and these are going to be used for drug screening and therapy of these patients in the probably, hopefully, in the near future. Okay, so this is based on a new approach, which is cell-based image analysis of cells coming from patients, which is the road. Why cells coming from patients? We believe that cells, non-neuronal cells extracted from patients from ALS, for example, can be a good model. I will show you evidence for that. That can be a good model not only to study the disease, but also to personalize drug screening purposes. Drug screening, personalized medicine is our aim. We have a facility in our lab that can do thousands of drugs, and we are aiming to have one drug, one pill that will be personalized for each patient, and one drug, one patient. And this is, our aim is meant to be for finding cure for those diseases like orphan diseases, rare diseases like ALS, which very low or very low funds and research have been taken by pharmaceutical companies to find a therapy or a cure for these diseases. So, the question is which platform and what is a platform? So as I told you, we are finding cells derived from ALS patients that are being considered disease models. I will show you evidence for that, and that means that these cells show traits of disease, which are different from cells that are not coming from patients with ALS or from people that are so-called healthy controls. The cells that we use are those that are coming, for example, from the bone marrow, mesenchymal stem cells, or other ready-made cells coming out from skin biopsies, like skin fibroblasts, which all have a proliferative capacity in vitro. So we could take one sample from the patient, grow it in our culture dishes, freeze them, thaw them, and also at all times we could get back the same results, and reproducibility is the name of the game. This makes these cells suitable for drug screening. And now I will show you for the first time that these cells can show disease phenotypes or biomarkers which could be identified or used for such purposes of drug screening. So what is high content screening analysis or image analysis using cell-based assays? So here we have a multi-titer well with thousands of cells grown in a dish. We stain them with specific dyes, put them under a machine, which is a microscope. We do high throughput imaging analysis. These cells, for example, are stained at different colors. The machine or algorithms can identify the meaning of these signals, and at the end we turn up with specific or numbers or graphs numbers that can identify differences between these cells, including difference in treatment, behavior, or between cell populations. And this makes us a perfect system for evaluation of high throughput screening of drug on disease based on phenotypes without knowing the molecular etiology. Basically, we photograph cells, they show us the biology, and based on this biology of disease, we could now start screening drugs without even knowing to which targets we are aiming. So this makes an ideal drug discovery on orphan diseases. So to make a long story short, the system, how it works, for you to understand the depth of this technology. For example, here we have a photograph of thousands of neuroblastoma cells, which makes networks that are kind of neurons. And for example, the system identifies each cell, and here are cells that are being labeled with 3 different dyes. Here it identifies each of the channels, because these are fluorescent labeled cells, and it gets until the very best resolution of making all this contact. This can even be quantified. So this is just for you to show you the complexity of this algorithm, how they are good to make interpretation of images. So how does it work? Basically, you have a cell, and within the cell you have different organelles. Whether you have a nucleus, a lysosome, a mitochondrial peroxisome, Golgi, etc., you can label these organelles one by one using specific fluorescent dyes, and based on the images taken from labels on these different type of organelles, you could subtract the specific information on each of these channels. One channel stains the nucleus. This is, in this case, is a peptide labeled showing which is introduced within the cell or not. You can measure that. You could measure the effects of any drugs on the cell population, even at the cellular level. What is the effect of a drug within the cells or outside the cell? And for example, here you have how it looks a year staining, a mitochondrial staining, which each has been analyzed by our algorithm system. So, just to show you in the first glance how does it look with just by having a bird's eye perspective, you can see, for example, cells that have been from ALS patients and control cells or healthy controls are being stained for different compartments, cellular compartments like nucleus, mitochondria, and so forth and so forth. You could see that there is a major difference, or kind of a difference between how these parameters are shown in multiparametric analysis. You can see that this, for example, the blue is different from the red, which is the ALS. We still don't know the etiology, why this is happening, but just as you can see in a kind of a very first glance, there is a difference. I will show you further evidence what the meaning of these differences. Okay, so here we are in the cell screening facility for personalized medicine at the life sciences faculty, where we have in our aims to personalize drug treatment of rare diseases. These are the diseases we are now working on, and we have here a robot, which is the liquid handling robot, the Incel 2000, which is the imaging system, an incubator, and everything has the facility to do a 24/7 application for drug screening. Here we have computers, a server, and everything is available for producing thousands and analyzing thousands of data. So how does it work? Our pipeline strategy works like that. So you have to bear in mind that we are not a pharmaceutical company, and our system is based like all diseases that we work with follow the following pipeline. So basically we have patient skin or human mesenchymal stem cells, fibroblasts, cells coming from patients. So we have cells coming from certain defined diseases, like in our case with ALS, for example. We identified phenotypic difference, whether there is a phenotype, a target, we are probably a known gene that's already known associated with the disease, or biomarker, which in a minute I will define for you what is a biomarker that is suitable for drug screening. So once we have this assay going, based on image-based analysis, we could expose these cells of each cells from each patient using a repositioning library of FDA-approved phase 3 library that we have in-house, or it could be a focus library, whether this is kind of a library that is targeted, or a naive library that we already have, which is unknown new molecules that can be detected for future drug development. On the third step, which is at this point a little bit farther away for us, it's a kind of validation, optimization, and doing target identification of the potential hits to make a lead compound for future drug or preclinical, clinical evaluation of those drugs, which is going to be done elsewhere. Okay, so to show you our feasibility of doing things, I will show you first screening we did with 10,000 compounds for fibroblasts with APBD, adult polyglucosan body disease, which is a genetic hereditary disease with about 70 patients in the world. Most of them are from Ashkenazi Jewish origin, and there is a mutation in an enzyme that degrades glycogen, and what is produced is producing accumulation of glycogen in brain cells. Miraculously, or for some reason, the symptoms of APD are very similar to ALS. Okay, so we have here cells coming from APD fibroblasts, from APD patients. We exposed them to 10,000 compounds, and we got 11 confirmed hits. So how does it work? Here we have a target. So we know we measured glycogen accumulation within the cells in red. The cells are in green, and you have here nuclei. Probably if we turn down the lights will be even better, but if you see it, it's okay. And based on, we found a hit. The hit will mean a drug that brings down the intensity of glycogen within the cells. And we already got 11 that were being confirmed using concentration-dependent curve analysis. And here we see that 11 that are behaving like they should be. And this was done in collaboration with Kahlon from Hadassah Medical Center, who is an expert in this disease. And based on that fact that we already know how to do screening, I now wanted to put forward the meaning that non-neuronal cells from ALS patients are good models for ALS disease. One, because they are showing novel biomarkers. We already proved and showed that 4 biomarkers, C1FIP2, RBP1, SLP1, and TDP43, are expressed in patients with ALS, whether these are coming from bone marrow mesenchymal stem cells or from peripheral blood leukocytes. And the strongest evidence that means that these sporadic or 4 genes that have been isolated from these samples has a meaning to the disease comes from the mouse model, which we have shown that there is the generative information, the generative progression. These genes are being expressed in the brain, muscle, and spinal cord of this mouse throughout disease progression. So, to make a long story short, these cells, mesenchymal stem cells, when induced with an agent that induces double-strand breaks, you see a decline in viability after 12 to 24 hours. Moreover, and this is coming from 5 ALS patients and from 4 controls. So if we look carefully, we found that autophagy markers, which because of time I cannot explain what autophagy is, but this is it is a cellular response that is been shown in ALS that has been exacerbated, that probably is involved in neurodegeneration. In this mesenchymal stem cell, if induced with induction of double-strand breaks, we have an increase in one of the markers, which is called LC321, and as well, we found by high content analysis an increase in the intensity of lysosomal compartment, which is known to be associated with autophagy. Now, if we block autophagy with a known inhibitor called 3-methyladenine, we see that we blocked this induced autophagy process, okay? Meaning that ALS mesenchymal cell is a good model to identify disease pathways mechanisms like autophagy. To make more specific, we already found that one of the biomarkers we have isolated, TDP43, which is already known to be a known genetic marker, a known genetic probably inducer of the disease in familial and sporadic patients. We saw here that a parallel concomitant increase of TDP in the nucleus has been shown and measured, that it is related with cell survival, which is known here. You see TDP43 in green. You see that after 24 hours, the cell density is going down. Okay, now in 1 minute, I will show you the best. So there is a characterization of ALS patient cell phenotypes. So we labeled different compartments, lysosome, mitochondria, cytoplasm, so forth, with different dyes, as I explained you before. And we found, and this is 2 samples from a multi-parametric analysis, in mitochondrial labels, you see a difference in the cell population of how they express this intensity, which is different between the 5 ALS patients compared to the 4 normal healthy controls, whether it is lysosomal or mitochondrial. Now, as I can show you, these 2 parameters are a set of 24 parameters that have been measured simultaneously on different cellular compartments. We have shown a significant difference from the normal mesenchymal stem cells control. Okay, what I'm showing you, this is a broad phenotype. Just by photographing the ALS cells, they show signs of difference, a phenotypic difference which is reproducible, and as well it is a product of a different disease mechanism which is built in the cells. This is, we haven't treated the cells is just growing at the same medium as the controls, and we found by analyzing the images a different, significant difference in each of these parameters between healthy and sick, which overall, this is a phenoscore analysis. What means we have a phenotype which is reproducible that says this is here ALS, okay? Which is, I don't have time to show you, it is different from other diseases. And now, to make to the bits of the more details, you could see in red to yellow, we have the ALS individuals. So this is a parameter showing exactly the same parameters I showed you before for each individual, which is significant difference how they behave compared to the normals, which are very close to 0. Okay, so this is the depth of our analysis. We can go from a population down to the individual and identify in the individual cells how they behave and what their phenoscore. Okay? So ALS patient cells show a disease cell phenotype that can be used for future diagnosis and personalized drug screening. Okay? So to make a long story short, and I would like now to summarize our ideas and why we think that personalizing ALS drug treatment is possible is because identification of ALS disease phenotype in patient cells by high content analysis. I've shown you evidence that this is possible. This could be done for development of drug high throughput screening assay, and this is what we are going to do very, very shortly. Screening of FDA for phase 3 and new compound libraries, which is going to be done. Identify potential drug compounds for ALS and the personalized level. This is also a possibility. Identify disease pathways and molecular targets is only our way, because if they show a pathway, these phenotypes are product of thousands of genes that are being showing a disease phenotype, and this can be identified. Drug development for treatment of ALS, and most important thing is this will be a platform for drug repurposing. Using FDA-approved drugs, we will do drug repurposing for ALS, which will solve money, will save time for drug development, and possibly in the future, drugs that have been FDA approved could be translated for treating ALS. Okay, so these are the people involved. Shane made all the experiments showed you today, and Brianna, Saja, Annie, and Eitan are past students, and this is important. All these technologies are not only developed by our group. Obviously, we need computer science engineers, known mathematicians, known biosystems scientists, obviously chemical computational chemists, and medicinal chemists from the Maryland University are part of our collaborators, and thank you very much for your attention.
So thank you for a very impressive talk, and I see a great progress since last year, which is very impressive.
Without money.
Without money, and also I think that you and Danny often should collaborate because of the phenotype, and I think you should talk more. So I'm not sure we have time for questions, only if somebody is on fire.
No, we have time.
Okay, Mira has a question.
No.
So do we have any question?
So I don't understand in terms of the lysosomes, also the ALS.
Do I need to say?
Yeah, you need to answer.
No, in the ALS fibroblasts, you found that lysosomal acidification is up or down?
It's up.
It's up.
And it's up, and it's going.
That's what we are saying.
It's going up. It's going up.
So you think
But
that
okay.
the question
No. And I want to.
the question if they do function, or it does represent some abnormal parenchymal.
Okay, just to remind you. Okay, so just to remind you, what I've shown with the increase of LC3, 1,2, and the lysosomal was induced with NCS, which is neocarcinostatin, which is in a double-strand break inducer. So this is a responsible double-strand breaks. So for some reason they die, and they die by kind of an autophagy-dependent manner. Yeah. What we showed that, Shane showed that there's a kind of a flux between LC3 1 to 2 is exacerbated to the 3 to LC3 to.
But the question, yeah.
The other, okay. So, yeah. So in the untreated levels, with what I've shown in the Pheno score, yes, we saw that difference that goes up or down in the lysosomal content.
What do you mean goes up or down?
There's defined.
Either that or that. You don't need to show us. Just.
No, we found it that untreated, most of the lysosomals are up. So what I showed is up.
How do you explain it? It's interesting
because what I think you have to bear in mind two things. We put
we
don't know if the lysosomes don't function. That doesn't mean that the intensity of lysosomes, or it increasing their area, doesn't mean that they are doing their job. Probably they are not, because they are dying. Yeah, but on the other hand, probably that something is stuck there, and there is the cells are more prone to show these anomalies. Whether these are functional or not, we don't know yet. But we are taking at face value the differences. Yeah.
So Teva representative, you don't. Okay, I think we should. Yeah.
The cells, I don't know if the patients are living anymore. The cells were taken many years ago, okay, and these were, we are continuing with our experiments. It's not taken freshly from the experiment, There are cells that grow independently of what the whereabouts of the patient. So it's not a one-to-one what is going on with the patient. They have been taken, and these are individual models of disease.
So he doesn't know.
No.
So I think we should continue. The stage of the disease is important. Also, maybe the cells that you are testing it in are important. These are. Exactly. So this is very interesting, and we should continue this discussion and work. And the next, so thank you very much.
Bye-bye.